So now it's my extreme privilege and honor, pleasure to introduce Fabian. And Fabian has been with us since 2015. Fabian is sort of legendary for both his independence and for his kindness. In fact, whenever I have a problem, he apologizes. So he's such a nice guy, it's been unbelievable. But Fabian got his, both his bachelor's and his master's in Costa Rica. He's a native of Costa Rica. And the one thing that he's really brought here that's a little different from what most people are used to hearing about is he primarily works on coffee and has always worked on coffee. So I guess with that, I'll let Fabian do most of the talking. I know he's got a very intensive seminar.
Thank you so much, Dr. Murray. So thank you all of you for coming here. It's really my pleasure to share a little bit of what we have been doing on coffee, and especially in this class, that is the coffee transcriptomics and some quality-related traits. So I just want to give like an overview. Many people you haven't been in a coffee farm, so this is what it looks when it's flowering. So coffee is a really important crop. For many people around the world, it's one of the most favorite beverages around the world, and even, for example, in the United States, almost a fifth of all the production is consumed here. So it tells a little bit of how many people really like this beverage. Normally, the production of coffee is done in more than 70 countries around the world, normally in the tropic area, and it's important for more than 125 million people. So it also tells a little bit of the distribution of the coffee and the importance of it. Of course, the economic value of this crop is really important too, and that's one of the reasons why we really want to have more production. So I want to just show what is important in the coffee crop, and the importance for us as readers is the plants. So we know that the coffee production is made especially by two species. However, we have more than 124 species around the world. Coffeacenephora, what that you call like Robusta, is one of the species that is grown, and it's around the 40% of the area. But also we have the Coca arabica, that is a special coffee species that we normally use, and it accounts for around the 50%, I'm sorry. So the Coca-Canephora is a diploid, and the Coca arabica is a tetraploid. And it came from the natural and spontaneous hybridization with Coca-Canephora and the Coca genoides around 0.6 million years ago. So it has the two genomes active working, and that makes a little bit of a difference that we have in the Coca arabica and the Coca-Canephora response. For example, for quality, we know that Coca arabica is known because of the quality. And normally the Robusta is used more for blends and caffeine extraction. We also have the more productivity with the Coca-Canephora. Also, we have more genes for resistance and more traits that we want to have from the Coca-Canephora. The variability is also higher in the Coca-Canephora, where we can exploit more that old processing processes. However, the variability in Coca arabica is really narrow. And that's one of the problems that we have right now, because around the 10% of all the wild varieties that we have in Coca arabica are exploited right now. So it's the 90% of those plants that are not exploited at all. So that gave us a lot of limitations. One of those is the most spread around leaf rot disease. So we call that Roya. And every place that is growing coffee, it has. Even, for example, I went last year to China, and in China you have leaf rot. And we have the limitation that we have a reduced diversity in Coca arabica, but also we are exploiting more the susceptible varieties that are what the market normally is wanting to have. We also have problems with a low profit or high investment, because to control these plants, we need to have a lot of management, especially chemical control. We also have climate change that is affecting all the crops, but of course also is affecting the production of Coca arabica around the world, since we are having different temperatures right now, and that is increasing the percentage of incidence of the rot incidence right now. So also we have more than 53 different races of rot right now, and more of them are getting like, are jumping the bridge with the resistant varieties. So just to make a difference or comparison, what you can expect from a resistant variety and a susceptible one. So as you can see, the farmers, when they saw these, the one on the right, you won't like to have that cultivar. However, it's the most common. And we don't have investment, we cannot apply any fungicide. So our control will be really bad. And we are going to lose yield, but also we are going to lose quality. So that's part of my project. I want to know how does the management, the genotype, and the disease are involved together in something that we can express like a disease. And how much that stress is going to be affecting the plant performance and the quality. So in order to do that, the plant performance, we can express that by the yield, or the number of leaves, or maybe the general condition of the plant. But the quality is also some kind of subjective and also tricky to work with. But we are trying to use near sensory analysis and also trying to find the volatiles by chemistry, like normal chemistry. But also, one of the most important parts to me is the RNA seq. We want to know the transcriptomics of this interaction. So why? Because we know that from the central dogma that DNA is going to be transcribed to RNA, and RNA is going to be translated into protein. Somehow, we are relating as breeders from a genotype to a phenotype, or sometimes we want to infer from the phenotype what happens in the genotype. And the leakage of that is here. And it's the largest amount of data that we have in the field. So we have in our analysis, we are normally exploring the transcriptomics in a type of tissue that belongs to the individual. And that also affects what we interpret from that, but also we know that environment is going to change by time and by condition. So in our analysis of transcriptomics, we know that we are subjected to this type of influence. So in order to explore this in my project, what I try to do is to evaluate two cultivars that are susceptible to leaf rot. However, one of those is the most common cultivar used around the world in some of them. Let's say that 90% of all the coffee grown is called Catuai, and a new hybrid that is a cross between the Catuai and another cultivar that wasn't being explored before. So this one is a little bit tolerant to leaf rot. We apply the fruit thinning. That is, it's not a practice that we use in coffee because it really costs, it's really expensive, and of course you're losing your yield. However, we will try to simulate what is the alternative bearing effect that normally we have 1 year that is higher in production, and we have the following year it will be lower. So we will try to simulate the same effect in 1 year and one condition, just to have that evaluation. But also we apply the chemical control as normally we apply in the management practices around the growing area, and to try to reduce the incidence of rot. But also we know that is a practice that normally is applied in coffee. So the last spray was done 90 days before the harvest was normal. So we will have all these interactions. What we did in the first thing, it was to reduce every other node the number of the fruits. So you can see what happens what you have in the plant before and what you have in the plant after that. That was done 60 days after the last flowering because you have different flowering during the year. Our experimental design was we had two plots, one from the hybrid, another one from the inbred, and we divided that one, each one, in the section that we are going to apply or we are going to spray that with fungicide, and the other part that we didn't apply any fungicide. After that, we also use the rows, and we have a fruit thinning in one of those, and another one not. And in that sense, we have like a six plot design, where we were trying to analyze 10 plants per repetition, but also 6 branches on each plant. So at the end was a lot of data that we collected for 6 months. What type of data we collected? And we're going to show you just a little bit of the results that we had. So we collected data from the rust incidence, rust severity, rust correlation, number of leaves in each branch, the general condition of the plant. That was a subjective evaluation between 1 to 5. So 1 really bad, 5 really good, and yield. It was collected by plant. So that's the measure of kilograms by plant. So you have all the interactions in here, but I'm sorry, it has to be a little bit lower. So at the end, what we were trying to say is that the rust control was the most impacting effect that we had in the control of the rust that we were expecting, but also it was for a rust incidence a 12% of control. However, for the correlation it was a 27%. And that's the most important one because we are going to avoid the new cycle of the rust. Fruit thinning and the cultivar or using the hybrid, we used around 4%. The incidence and wasn't so big the difference for correlation. That was our effect. Those were our effects in the plan. However, how about the quality? We were interested about the quality. In order to do that, every time that we harvest, we were collecting the fruits. However, we used the ripening or the best moment or optimal moment when we can harvest the fruits. So for each one, we were trying to collect all of the treatments and reps. That's the quality of the coffee. Later, we transferred that to the mill where we wet processed that. We took out all the pulp, the parchment and all that. We dry it, and later we have what is called a cold bean. Those were packaged in vacuum sealed. So we had 4 biological grades from every grade that I had in the farm, and I just packed it in 300 grams. 300 grams, and vacuum sealed them to take them in here to the United States. We did those samples were taken to a place that they roasted them, and they also made a sensor analysis that is called the specialty coffee oxidation analysis. I'm so sorry. Okay. And with that sensory lexicon, they used to have a description of the body, the acidity, the preference, aroma, other type of attributes of the coffee crop. And they also gave the same sample to make another protocol. That is a new one that is called the World Coffee Research Sensory Lexicon. That is used like an attribute for example, chocolate. How much chocolate I have, how much intensity I have from 1 to 15. So you have also multidosity, you have cell aroma, so they have different descriptors. And this is just one of the things that they were trying to improve. So after that, the same sample that was evaluated by the World Coffee Research Lexicon sensor analysis, we also, with the help of the people in food science, they helped us to make the chemistry. So we were using solid phase microextraction, gas chromatography, mass spectrophotometry to analyze that. And it's really funny because I was smelling all the aromas of coffee. Something that normally, you have the beverage, but you don't realize all the aroma that it has. But when they separate that, you realize all the different types of aromas. So I'm just going to show you one of the descriptors or profiles that we were using. So as you can see, every dot that you have in there, it means 4 reps, and it's H value that you have in here is one attribute. So it will be, for example, an S means that you have a sensory that is, for example, chocolate, and those are 4 reps used to make this mean. But also you have the volatiles, and we obtain at the end 60 sensory attributes, and we obtain 154 volatiles. From those, just 10 attributes were important to us, and from the volatiles, just 18 were important to us to understand what was going on. But you can see here that there was some clustering. Those points that were correlated were also associated with the treatments that were less stressed or more stressed. And that's one of the points that we were trying to find. We have profiles in the coffee because of the treatment. So the management is affecting that. And you can see that more stress factors are clustered in the right side. These ones are in the left. But what is interesting is that we had, I will just explain two of those volatiles. That is the V5 and V82 that are on those squares. And we are going to follow the history of those. So we had a lot of correlations from the volatiles and the sensory parameters or attributes, but just the V5 and the V82 were the highly significant correlated to different traits. Here I'm trying to describe, for example, the overall condition was negatively related with the rot incidence, and it's minus 0.85. So when you increase the rot incidence, you have less overall condition. So it is logical. You also have that for severity. So we have, for example, when increasing rot severity, you also have an increase of the rot incidence, and the correlation is really high. What is this and this, as you can see, these volatiles, this was a V82, that is acetaminophen. It's the first time that is reported in coffee as a product of a roasted process. And this other compound that has a large name is a naphthalene. The naphthalene is a precursor of many aroma compounds. And what is interesting is that we found that this volatile was negatively associated with a harvest, and acetaminophen was highly correlated with the rot severity. But that was highly significant. But I want to show you that we have other significant, like 95% of precision or confidence in other type of compounds and another type of attributes. So it's really interesting, and we report that in the paper that we already, it is really recent. But what we want to know is the gene expression is also altered because of these conditions. So, well, that was thanks to all my committee members for doing and the help. So, to do that, we have to explore the gene expression. How we did it. So, all the samples had to be collected and processed in the same place. So, we went back to my university in Costa Rica. So, they gave me a full stage where I put all my things. Later, I had to travel with nitrogen, a lot of nitrogen, to the farm. It was a 2-hour travel to go there. And I had to sample, I did the same thing for these. But of course, well, we are interested in the coffee and the fruits. So I collected two types of fruits, immature and mature fruits, from each of the treatments. I took the fruit out, just put it inside of a plastic bag, and later placed it in nitrogen, and later traveled with that to the university where it was stored at -80. So I started to process that. Of course, I was trying to simplify the things using a grinder. I destroyed that one. So I continued doing the most practical way, mortar and pestle. Okay? And that was part of the batches that we were trying to find. And when I was doing the analysis of the quality, I was really happy. Really good concentration, really good quality. However, when I took the samples in here and I analyzed them again, the quality wasn't good. There was a lot of degradation. So the problem that I had, several problems that I had, was that we were needing new reagents because the ones that I had before, they weren't in good condition. So, and we also used this spectrophotometer that didn't give me the 260 to 30 ratio that estimates the concentration of salt. So there, with the help of Barbara, we arrived with a new spectrophotometer. It is the FabianEcheverria spectrophotometer, and we have really good concentrations, really good quality. So we continue forward. And after 12 days or 2 hours of drinking, we got the samples, and also we tried to store those samples in some way, or we were trying to make them in a easy way to transfer in here, because we were using before an alcohol, and the alcohol didn't work really good. So we were using this type of the second pellet that we were trying to transport them in normal conditions. So after that, we sent the samples to the sequencing facility. It was in Cornell. They helped us to make the Lumina sequentiation in HiSeq 2500. And later, all the data was analyzed in here. Dr. Klein helped me with the trimming and adapter removal. But later, I followed the next process where I was trying to align the sequences to the genome that is available, that is a copy of an FabianEcheverria genome. And I was trying to find the expression values, which genes are important to us, and which treatments are important. So I followed the whole pipeline. The sequenciation was good, and the quality was really good. We obtained at the end 88% of all the reads were mapped to the genome in the section of, or were mapped to the gene. And when we were trying to make the analysis of how many genes were expressed because of the sample, we obtained that 35% of all the genes that are annotated were transcribed. I'm sorry, they were expressed in the field. Yes? Yes. Yes, sir. Sorry. So we were trying to find, at the beginning, which genes are really differentially expressed because they were in the immature or the mature stage. So we made this analysis where we found that 1,000 genes were differentially expressed because they were, for example, more expressed in the immature, or they were more expressed in the mature. So as you can see, we have like 6 times more differentially expressed genes in the immature phase, where we were trying to have an analogy of what is going on. So are they functionally relevant to us? So that's the next step that we try to do. So we try to make, from all those genes that are in the immature phase, which ones are, we can categorize them as functional. So we did a gene ontology analysis using AgriGo, and we have different expressions and enrichment in different categories from the Go analysis. So we have metabolic, catalytic, transportation, redox, but still, what we were interested in, from where? So it seems that using the Kyoto Encyclopedia of Genes and Genomes, they were more associated to pathways of the biosynthesis of carbohydrates and fatty acids. And that was really interesting. However, those ones are associated to stress. We were following the same logic, and we found like 211 differentially expressed genes that were expressed in the immature stage. In the inbred, we also have another set for the hybrid, but we were interested to know if those ones were also affected by the treatment. It means by stress. So I'm just setting 10 of those that were also clustered in the Go category, and I'm just showing you here some of the genes and the fold change. So it means that when you have downregulated genes, it means that the expression of those genes are according to more stress, while if they are upregulated, you have more expression when they are in less stress. And these 10 genes are interesting for us because of they are going to be associated with something. Is our prediction. We were trying to know if that happens. So, for example, in this case, I made some stepwise regression to verify if the expression values of those genes can be also related to some compounds or some traits. This is just a model. Of course, the R-square is not really high, but I'm just considering just one gene, that is the stromal cell-derived factor, that it was taken by the system, by the analysis that we did. But the interesting part is that these genes in Arabidopsis, in mouse, and in humans have been found related to the endoplasmic reticulum and is expressed during stress. So it seems that our approach was like is getting into something that we can use, and that's more or less the goal of this. So we can say that this is a candidate gene that, of course, somebody else will have to take it and validate. So far, from our experiment, we are getting into the analysis of what is the importance of those genes and the precursors of the quality of the coffee. So from this analysis, we are having some data that is showing that in the mature phase is where almost all the anabolic processes are made, and in the maturity stage, we have a process of more catabolic, so it's a transformation of those compounds, and combined probably is what is giving us all the expression of the volatiles and sensory analysis. So far, from all the analysis that we have been doing, we can say that we can integrate to this model the post-harvest process. So all the post-harvest can be affecting these interactions, but also we found that some of the plant performance traits that we can say they are some positive, so they are green, or negative traits like rot incidence, rot severity, can be affected, but also how does the quality can be affected. So we have some traits, for example, in the sensory, and we have in the chemistry part some volatiles that were also affected. And we had some of the transcriptome analysis that some carbohydrates, fatty acids, and those pathways are related also to the synthesis or processing of these. So to my goal is to find at the end if those ones are correlated. So we found, for example, overall condition is correlated with acetaminophen. When you decrease overall condition, you have more acetaminophen. When you have more yield, you increase the acetaminophen. I'm sorry, the overall condition was opposite. Later, when we had, for example, more rod phosphorylation, we had more cell aroma. When we have more rod phosphorylation, we decrease the overall condition, and so forth. So we had also the fatty acid and volatiles, and for all of them, we had some correlations or idea of where they are going and what is the interesting part of them. So why we want to control them, to use something productive, something that we can use it, and of course explain why we have to protect a plant, why the disease is important to us, and of course if we can improve this model and make predictions, we are going to avoid a lot of years of exploration in the coffee. For example, we need like 25 years to breed. So that would be really, really helpful. So as you can see, well, it was a really multidisciplinary project. So I really thank all the people that are involved in this, especially my committee members, and the funding from USAID, from the World Coffee Research, and all the people that have been helping me with all the processing, the molecular part, the sensory part, all the chemistry, and of course, in the Burlock and the core lab as well. So I really thank all of them, and also thank all of you for your attention. So I guess we have some time for questions if you have.
One of the questions, and I'll point out one thing that you kind of mentioned that I think was really surprising to me, and you were the first person to find it, which is that acetaminophen, was produced in coffee under stress.
That's correct.
And so you actually published that already, and I think it's pretty exciting. I mean, compared to 500 milligrams of 1,000.
Yeah, so that's one of the problems that we have in the volatile, and I was asking for like more budget to make the other side of the chemistry, and they denied. But that part of the chemistry, for example, I'm storing more in the hybrid, and all the interactions are getting me into the point where, okay, the phenylpropanoid pathway is really related to that production, and since a lot of fatty acids and all of compounds are related to that, probably we can get closer to a really good explanation of that. But yeah, it's kind of like exploration processes, but we have to confirm and validate. Like you said, we need to know how much exactly was in there.
So if you drink coffee and alcohol.
Yeah.
Oh, yeah,
Yeah.
exactly.
That's right.
Yeah, what's the, how are coffee producers paid? What's the most valuable measurement for paying coffee producers?
So,
On yield or they paid on quality?
so normally they pay by both. However, the massive part of coffee is paid by just the stock. And for example, just making a comparison, in Costa Rica, the cost of production of coffee is $0.53 per kilogram. The cost the price that you receive is $0.63. So you just have 8 cents per kilogram produced. If you transform that to what the cost of for example, a glass of coffee, a whole one, is 1.85 in some places. And the thing is that what you need to produce that or to prepare the beverage is 2 cents from those 1.83. So the quality is really not like valuable as a service.
I was wondering why it was, because in your results, you showed that rust had no effect on yield.
Yeah, so in our experiment, what we obtained is that it was a small difference at the end per plant, and if you transform that to hectares, it is a really small quantity. However, it was a really good, like, agronomical condition, it was good. If you keep that plant without all the fertilizer or all the patches that you need to control it, you won't have any yield next year.
So my final question then was, the other thing I took from that slide was that your yields increased substantially with the hybrid.
Yes, correct.
So how many people grow hybrids, and if you're not growing hybrids, how come you're not growing hybrids?
So normally those hybrids are not like the crosses. They are clonally propagated. So the cost of that plant is twice what costs a normal plant.
How long does it take to produce?
Pardon me?
How long do you have been producing trees?
So yeah, we have plantations like 30 years. Yes. So if you compare the investment that you make, at the end you should change for hybrid because of the production and probably with different qualities, because this is just one of the hybrids, but of course you have another type. And however, the producer is more skeptical sometimes. Also, the level of production of that hybrid is is more stringent, so they don't have like a really good market where you can get those hybrids in an easy way. And yeah, those are part of the con. But yeah, that's why I prefer like the hybrid technology is going to be something important, but the producers is really important. I'm sorry. Thank you.